Hi everyone, I'm Caroline with Fiveable, and tonight we're going to do a little intro to AP biology. So let me flip my screen really quickly. So my intro is targeted to new, current, hopefully starting out fresh this September AP biology students. So it'll be relatively brief tonight, maybe about 30 minutes. And I'm just going to run through a couple key topics that are really good to know and have in your back pocket going into AP. So nothing that I talk about tonight will be at the AP level because we will get there as the year progresses. This is just kind of the background information that you should be familiar with. It's especially great if you haven't taken a if you haven't taken a biology class before, but if you have taken a biology class before, that's wonderful. Hopefully we're just fine tuning, dusting off some cobwebs here, and getting you ready for a great year. So before we jump in officially, really excited to be working with a team of bio teachers this year to bring you some awesome AP biology content. We are going to go through absolutely everything that you need to know to be successful for this course. And I'm really, really excited to have a team of teachers and students interacting with you all throughout the course of the year. So this is just a very, very small intro to what the rest of the year will be like. And again, it'll be about 30 minutes and just going through some stuff not at AP level, just kind of some background biology. All right, so coming soon, just to kind of get you interested and excited for what we have to come, our first unit, which will probably be your first unit in the classroom as well, is the chemistry of life. So we'll have sessions on the structure of water and hydrogen bonding, the elements of life, and then lots of stuff about macromolecules, all of those chemical things that make us up, some of which we'll talk about tonight. In this stream specifically, here are the main topics that I'm going to go into. So a little bit of chemistry, because that's going to be our first unit, a bit on cell structure and function, some heredity, gene expression, and evolution. So again, if you've taken a biology course before, these are going to be some of those key concepts that you know and are bringing with you. If you haven't, we'll just get some vocabulary in there, and you can always rewatch this video if you forget before the start of the school year, or if you get confused, just for some of that introductory vocabulary that your teacher might expect you to know. Okay, so let's start by first talking a little bit about chemistry. So the three major things that I kind of wanted to review with you all tonight is the structure of water, bonding, and macromolecules. So depending on how your school does AP biology, you may or may not have had chemistry beforehand. It is really great if you have had chemistry before AP bio. My students haven't. I didn't when I took this course. You will be just fine, even if you haven't had chemistry. But there are a few things that are helpful to know going into it. So the first one is the structure of water. Water is the universal solvent. It means that so many different things can dissolve within water, and it has a lot of unique properties. Without those unique properties, nothing on earth would be alive, okay? And I can talk to you a little bit about that as well. So the structure of water you can see on the left-hand side over here, H2O, if you're familiar, 2 hydrogen atoms, 1 oxygen atom. But the really, really important part about the chemistry of water is that it is a polar molecule. So you can see that word polarity here. So polar means that the electrons that are housed within this molecule of water are not shared equally. So something that's important to know going into AP Bio is that oxygen is the most electronegative atom on the periodic table. Electronegative basically means that it's really, really greedy for electrons. Someone once explained it to me as Gru. If you're familiar with Despicable Me, he's very negative. He loves negative things, at least in Despicable Me 1. There's like 17 Despicable Me's at this point. I don't know what Gru is like at this point, but he's really, really negative, loves negative things. That's oxygen. So oxygen is very greedy for electrons. And you can see that in the structure of water on the left-hand side with that little negative charge above the oxygen. It's because the electrons from hydrogen are being unequally shared. They're being pulled closer to oxygen, which is giving it a slightly more negative charge than those positive hydrogens. Okay, so that molecule is considered polar because of that unequal charge. And that is going to lead to a lot of conversation when we start talking about the plasma membrane, which is what coats all of our cells. We're going to talk about the polarity of water. The other really unique thing that water can do is is hydrogen bond. Okay, so a hydrogen bond is going to form between the hydrogen of one water molecule and the oxygen of another. You can see my two fists kind of coming together. I'm very small down here in the corner. But those attractions, the negative of one oxygen with the positive of one hydrogen, form a really nice bond that allows water to do really cool things like climb all the way from the roots of a tree up to the leaves of a tree. If you think about the fact that water has to climb all the way from the roots up to the leaves, that's an amazing property of water. And those are some of the things that we will talk about towards the start of our school year when we start officially streaming the week of Labor Day. But something good to know coming into it, that water has a lot of these funky properties because of the polarity of it, which you can see on the left-hand side of your screen. All right, bonding. Okay, so a little bit about bonding. And Sarthak, I see your question. I have to actually double check because that may only come with a 5able Plus membership. In fact, I believe that only comes with a 5able Plus membership, but I will get back to that question for you by the end of our session tonight. Okay, so next, bonding. Another good thing to be familiar with. If you've taken chemistry, you're like, why are you telling me all these basics? That's a good feeling if you have that feeling. If you haven't taken chemistry, again, we're just going through the very basics here so that you can get through those first couple of weeks with a little bit of an advantage. So two major types of bonds that will come up in AP biology, because there is a lot of chemistry embedded in this course, is the difference between a covalent and an ionic bond. So a covalent bond I have up here is where we're sharing electrons equally. Let me change this to a pointer. Cool. Change the slide. Technical difficulties as always. But we're sharing electrons equally, and you can see this carbon molecule in the middle and then these hydrogen molecules. And you can see that you have an electron from hydrogen and an electron from carbon filling in this bond. These covalent bonds are really, really strong, and they don't dissolve in water. So usually nonpolar, things that share electrons equally, which is what you can see here, molecules are going to have these covalent bonds. And if you throw a sugar molecule, for instance, that would be an example of a nonpolar molecule, into water, it will dissolve, but the actual bonds, those covalent bonds that are holding the carbon and the oxygen, or the carbon and the hydrogen and the oxygens together, are not going to dissociate. They're not going to break. So they're very, very strong, and they happen in nonpolar substances. On the flip side, ionic bonds happen usually between two atoms that are unequally matched. So sodium has a positive charge and chlorine has a negative charge. They love to bond together. NaCl, sodium chloride, is table salt. You eat it every day. We put it on our food. Okay. If you dissolve salt in water, okay, that bond actually breaks down, and we have free sodium ions and chlorine molecules floating around, okay? And you can see this here. These two are technically bonded together, but the electron from sodium is hanging out over here, unequally shared between these two things. And you can see those charges. We call those partial charges. And this is actually an ionic bond. So it happens between ions. I-O-N, the first part of that word ion, means a molecule that is charged, okay? So this is a sodium ion because it is Na+. This is a chlorine ion because it is Cl-. Things that have charge we call ions, and therefore ionic bonds happen between these charged molecules. Okay. And then the next major part of chemistry that's just good to know before you enter into AP biology, and again, you may be familiar with this from a prior bio class, are the macromolecules. So macro meaning large. I'm always going to break down things with root words because macro evolution is like large scale evolution. So we're going to see these things over and over again. Macromolecules are large groups of molecules, and they're the four molecules that make up everything that is us, that is living. Okay, so the four major categories that you should be familiar with going into this class: nucleic acids. So nucleic acids, which you can see here, and I hate that this little situation is popping up because then you can't see the fun cartoon. So I'll just stop moving my mouse really quick. Nucleic acids, which you can see right here, are what make up DNA and RNA. So we have two different types of nucleic acids that are made up, you can see the little word there, of little nucleotide building blocks. Okay, so DNA and RNA, which we will get into fully when we break into all these macromolecules in Unit 1, are in the nucleic acid family. Then we have carbohydrates. They're made up of monosaccharides, which you can see on the bottom there. Carbohydrates are sugars, all of our sugars, like things that I like to tell students, like pasta, bread, any type of sugar, any type, like fruit is high in carbohydrates because it's high in natural sugar. All of those things fall into the carbohydrate family. They're often times used as well for structural elements, which we can get more into when we get into the carbohydrate family, but they do a lot of cool things, and they're made up of little building blocks called monosaccharides. Next are the lipids. Lipids are made up of fatty acids. We can see the little fatty acids here, these little blue wormy guys. And this is a specific type of lipid. Lipids are anything like the plasma membrane that are going to have fats in them. So you would consume lipids if you consume oil or butter, or there's fat on your meat or things like that. And these are going to be hydrophobic. They're scared of water. And you can remember that because if you think about it, oil and water would never mix because they repel one another. And so therefore lipids, which are oil is just an example of, are going to repel water. And that's going to become a really important concept for you to remember when we start getting into the structure of the plasma membrane, which is one of the most important things that we will talk about this year. And then lastly, we have the wonderful proteins. So protein that you can eat would be things like eggs and meat. Proteins are really, really important. They're coded for by DNA. We'll talk a little bit about transcription and translation tonight, but they're built up of building blocks called amino acids. And again, the structure and the components of a protein are coded for by our nucleic acids. So there's a strong tie between nucleic acids and proteins. And we'll talk a little bit more about that as we jump into cells. Okay. Cool. So again, that is very, very brief overview of chemistry that you might need to know. Our first unit goes into chemistry in the extreme. But this is, so Sarthak, we should be good is kind of a vague question that I can attempt to answer. This is just the very much the basics of chemistry that you would need to know coming into AP. So none of this is AP level content. The AP level content will get started when you start your school year, or if you haven't already, and past Labor Day, where we'll take this and we'll put it to the next level. These are just kind of like if you've taken an intro bio class, these are the top things that you should remember from that class. Okay, so briefly with cell structure and function, and I won't go into a ton of detail on this slide, but coming into it, and again, the reason that I picked these slides and the content on them are because when I'm getting ready to teach a unit or teach my students, these are the things that I'm kind of expecting them to know. So I'm expecting them to know all of these organelles, and I'm expecting them to know what they do so that we can build off of them and talk about the more complex things about them. So that's why I'm presenting them as such. So just a brief rundown of organelles that you should be familiar with entering your class. The mitochondria. So the mitochondria, the powerhouse of the cell, the one thing that everyone learns in high school. But the mitochondria is that powerhouse. It looks up here like a little bean, and it has all these membranes in the inside. Those inner membranes just increase the surface area of the mitochondria because that membrane is where the electron transport chain for cellular respiration is located. If you're not familiar with the electron transport chain and cellular respiration, do not fear. That is never something that I expect my students to know coming into it. And we will have multiple modules on all of the complexities of cellular respiration. All you need to know is that the mitochondria makes lots and lots of cellular energy. We call that cellular energy ATP, and it is our little powerhouse in our cell that is powering us up so that we can do all sorts of cellular things. Okay. Lysosomes. So lysosomes are over here, these little pink guys. So lysosomes are really, really cool. Lys, L-Y-S, is going to mean to break. So a lytic, something that's lytic or lysing, all those things with a L-Y and an S or a T, that means to break apart, to burst, something like that. So that's a good prefix to be familiar with. It'll help you remember. So lysosomes are full of hydrolytic meaning breaking down with water, enzymes that are going to break down lots of different things in our cells, like old organelles, things that are dead, things that are damaged. They're going to break down like little recycling factories and then either dispose of or give back to the cell whatever's left. So they're really, really great. If one pops, this is like a question on the AP exam sometimes, those enzymes are going to be really damaging to the cell. So we want to keep them contained within the lysosome. Nucleus over here, big old guy, houses our DNA, keeps it safe. DNA can't leave the nucleus. We'll see why that's important to know when we talk about transcription and translation briefly, but it's going to do a really nice job of protecting our DNA, and it is the site of transcription, which we'll talk about in a second, which is getting our DNA into messenger RNA, which is then going to go be turned into protein. Vesicles are important. So vesicles are just like little bubbles. How many vesicles on here? Here we go. Little bubbles of membrane. So you can see that this is just fusing with the external membrane here. And a vesicle is just like a little vehicle. So I always remember vehicle, vesicle. So they're just going to move throughout the cell or out of the cell, and they'll deliver things or release things into the environment or out of the environment, whatever it may be. But they're just going to carry things around in the cell. They're like our little messengers, our little trucks. The rough and smooth endoplasmic reticulum are right here outside of the nucleus. The rough endoplasmic reticulum is called rough because it is dotted with ribosomes. So ribosomes are our little protein factories. That's where proteins are made from our messenger RNA, which I'll talk about in a slide. But the rough endoplasmic reticulum, which is covered in these ribosomes, is going to be our big protein production. Okay, and then smooth endoplasmic reticulum is more involved with producing lipids, so those fats and steroids and other things that are needed by the cell that aren't proteins. But both of those are going to be big production areas. And the reason that the rough endoplasmic reticulum is right after the nucleus is because we're going to take the messenger out of the nucleus and right into the rough endoplasmic reticulum to be translated, which we'll talk about in a brief moment. The Golgi complex is also nearby, and the Golgi looks like kind of a stack of pancakes, and it has a fun name, so it's a favorite organelle of mine and others. But you can see it here, and that is going to be involved in synthesizing our proteins and kind of fixing them up. So after they get made by the rough endoplasmic reticulum, they're going to go to the Golgi to be processed. So sometimes they get snipped, sometimes they get folded, sometimes they get a sugar slapped on them, but they're going to make them really fun and pretty in the Golgi complex with the fun name and the pancake shape. Ribosomes we talked about. Vacuoles are storage. You can see I put both of these cells here for a reason. This is the vacuole of a plant. It is massive versus the vacuole of an animal. Major reason there is that our vacuoles in our plant cells are going to hold water. So plants need to be able to retain water way better than animals do, because we can go grab a drink at the sink and the plant has to stay in the ground until it rains, right? So those vacuoles for plants are going to do a lot more than they do for us, but they're big storage areas. And then lastly, the chloroplasts, you can see them in here. They have some pancake structures in them too. We'll get to that. I love the chloroplast, but that's going to be the site of photosynthesis. So it's in any photosynthetic organism like a plant or a photosynthetic bacteria. And we will go through all of the complexities of photosynthesis and all of the enzymes involved and electron carriers, et cetera, in our energetics unit when we talk about cellular respiration as well. So that's just the brief overview. Again, these are organelles that you should be relatively familiar with going into the course so that we can apply concepts on top of that. A lot of people say bio is memorization. AP Bio in 2019 is not a lot of memorization. It's a lot of application, which tends to be harder. So you need to know the basics so that we can apply it to a new type of question. And that's kind of what we're up against. So that's what we will prepare you for as the year goes on. Okay. All right, cool. So far we have talked about chemistry. We have talked about cell structure and function, the basics. Now I just want to give you some vocabulary that you should maybe be familiar with going into a heredity unit. So just some things to know because they're things that my students sometimes struggle with. So here's our cell. And within our cells and within human cells and most eukaryotic cells, our DNA is wrapped up really tightly into a chromosome, which is often depicted as an X, which is somewhat deceiving because this is actually a duplicated DNA, but we'll get to that when we talk about heredity and cellular cell replication. But our chromosomes are just that compact wound up DNA. So if you unwrapped this, you would get lots and lots and lots and lots and lots and lots, like 2 feet of DNA in each of your little teeny tiny cells. So a teeny tiny cell, which we can't even see with the naked eye, has 2 feet of DNA in it, which is absurd. It's just wrapped up that tightly into chromosomes. So what you can see here is that lovely classic double helix ladder shape of DNA. And then our chunks of DNA have what we call genes in them. So genes are just a specific segment of DNA that codes for a specific protein. Okay, so something that's important to know that will kind of give you an edge above the rest, above the competition coming into AP Bio, is that the reason that we look a certain way, right? The reason that we have the color hair we have is because of a gene that we inherited from our parents. But more importantly than that, DNA doesn't do anything. DNA sits in the nucleus. It's not allowed to leave, right? So it's not turning your hair brown or blonde or black. What's doing that is the protein that's produced by that gene. Okay, so we're going to talk about that on the next slides, transcription and translation. Okay, but that gene, that segment of DNA, doesn't do anything. It just codes for a protein that does the thing. So if you know that coming into AP Bio, your teacher's going to be so very thankful, and everything's going to make a whole lot more sense, especially with heredity. Okay, so that's the first thing to remember with this kind of stuff. But that also goes when we talk about evolution and things like that. We'll talk about genes a lot as well. Okay, then the other kind of confusing vocabulary that sometimes students get confused about is this word down here, allele. Okay, and an allele is just a version of a gene. Okay, so I'll give you an example because sometimes students get a little confused, and especially depending on where your teacher starts teaching in what they start teaching. Sometimes they just like hit the ground running and they're like allele, gene, blah, 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 and students don't know what you're talking about. So an allele is a version of a gene, and we have 2 of them because we have 2 versions of the same gene, one from mom and one from dad. Okay, so that allele could be dominant, which in this case we represent with a capital letter, capital B, or that allele could be recessive, lowercase b. So what does that actually mean? Well, in this specific example that you're seeing on the bottom of your screen, a dominant allele means that that segment of gene produces a protein that turns flowers purple. Okay, so that segment of DNA is producing a protein that leads to purple pigmentation. Okay, so if you have that allele, whether you have 1 or 2 of it, doesn't matter, you are showing up as a purple flower. Okay, reason being that segment of DNA is producing that purple pigment. And whether you have 2 segments of it producing that purple pigment or 1 segment of it, you are still producing that purple pigment, and you are going to be a purple flower. Okay, so that purple color, we call that a phenotype. So I'll write that in here. Okay, so a phenotype is the physical appearance that you look at and can see. So in this example, it would be purple. Okay, so the dominant phenotype is purple. Okay. On the other hand, if you don't have any dominant alleles, if you have 2 recessive versions of that allele, then your gene does not produce purple pigmentation. You do not get that protein that makes the purple pigment. Okay. And you don't have either gene that makes purple pigment. So instead, you're a white flower, and we would consider that the recessive phenotype. Okay. It only happens if you have 2 recessive alleles. So the recessive phenotype is white, okay? And you can see that on this slide, Okay, so those are phenotypes, physical appearances. Now, I've already shown you this and you can clearly see this. We have some other words written down there. Those are considered genotypes. And genotypes are the genes that lead to the phenotypes. So it's what your DNA is actually coding for. So in that case, we actually have a difference between this purple flower and this purple flower because there's different genes that these two flowers have. On the left, we call this homozygous dominant. So homo means same, okay? Two of the same. And dominant because it's the dominant allele. So they have two of the same dominant alleles, big B, big B. We consider that homozygous dominant. They also have the dominant phenotype, which is that purple color. In the middle, hetero means two different, okay? Two different types. So heterozygous, we have a capital B, a dominant allele, and a lowercase b, a recessive allele coming together. And that is still leading to the dominant phenotype. Why? Because we have that dominant allele producing that purple protein, okay? But on the low, this guy's carrying a little secret. He has a recessive allele that could be passed on to his offspring, okay? And then lastly, on the right, we have two recessive alleles. So homo meaning same, homozygous recessive, two recessive alleles leading to the white flower phenotype. Any questions about phenotype, genotype? Maybe y'all are experts out there, but this is again something that sometimes students struggle with. You don't have to be perfect at this going in, but it's nice to have just heard the words before so that when you hear them again, you're like, oh yeah, I know about that. Because then we're going to take it and we're going to run with it. Okay, great. So then on the lastly on the right-hand side, and I'm not going to get into this, I'm not going to go through Punnett squares and things today, but you might be familiar with them, is just that exactly as I was saying, so between a mom and a dad, they obviously have a genotype, so two different versions of an allele. So in this case, both mom and dad are heterozygous because they have a dominant and a recessive allele. And there's a 50-50 chance that each of these individuals will pass each of their two alleles on to their children. So the mom could pass on her dominant allele to her children, or she could pass on her recessive allele to her children. It's a 50-50 chance. Same with dad. So because of that, you can create this thing called a Punnett square where we have mom's dominant and recessive allele, dominant and recessive allele, and dad's dominant and recessive allele, and we can see the different possible combinations of their offspring. Okay, so they can produce a lot of different offspring with their individual genotypes in this cross. So I'm not going to go through all the logistics of a Punnett square, but just know that both mom and dad have a 50-50 chance of passing on each of their two alleles onto their offspring. So that doesn't really matter if you're homozygous dominant or homozygous recessive, because the only thing that you are able to pass on to your offspring is either a recessive or a dominant allele. But it certainly matters if you're heterozygous, because you could pass on a dominant or a recessive allele onto your offspring. Okay. All right, so up next, and again, we're all over the place. We're going through 5 of 8 units and just going through basic things. So we're really covering the map of biology content tonight. But up next, a good thing to be familiar with before you go into AP Bio is the processes of transcription and translation. So we call this the central dogma, going from DNA to mRNA to protein. You should know that, you should love it, you should internalize it, do whatever you need to do, but put it in your brain that DNA is going to be transcribed, and we're going to make a copy of it called mRNA, and then we're going to translate that copy into protein. And we can get into all of the logistics behind the naming of those things at a different time when this is our top focus, probably in January. But you can see a little diagram down here that just shows this, and it helps to make sense of what I was talking about in the previous slide with the fact that genes don't really do anything. It's the protein that the gene codes for that does the work for us. Proteins are the workers. Proteins are the coolest. They're the things that matter the most. So what you can see here is we have our DNA, and we're going to make a copy of it. And the copy that we make is going to be RNA, specifically mRNA, and the little m stands for messenger, okay? And RNA, if you're not familiar, is very, very similar to DNA, except that it's single-stranded. So you'll see DNA has these nice 2 strands. We call it the double helix. Okay, it's single-stranded and it has a different base. So instead of having a thymine or a T base, it has a uracil or a U base. And if you're not quite sure with how these things pair up, that is a-okay. We're not going to get into all the nitty-gritty today. But what you can be familiar with is the fact that we have this DNA and then we make a copy of it in a single-stranded form. So you'll see that we're actually pairing up bases with our template DNA in this new slightly different compound called RNA that's hopping off here. This is our messenger, okay? And the reason that we have to make a messenger, if you were paying attention earlier, is because DNA can't leave the nucleus, okay? That doesn't do us a whole lot of good because all of our little protein factories, the ribosomes, are not in the nucleus. Okay, so we can't make protein from that DNA if it's just chilling in its little castle. Okay, so we make a cheap message, something that we don't care about quite so much, right? Like our DNA is everything. It codes for everything that makes us us, okay? But we can make a cheap copy. It's very, very similar with some slight differences. And this lovely thing can leave the nucleus and will leave the nucleus and go and get translated, okay, into protein. So you can see that down here. So we're going to read this mRNA at the ribosome and turn it into protein by pairing up specific amino acids based on the code that we're reading from this mRNA transcript. So I have a bigger image of this on the next slide, okay, so that you can kind of take a peek and see where this is all going. But again, know that you don't need to know all the nitty-gritties of this. You should just be relatively familiar with these processes. Okay, so on the left-hand side, again, we have transcription. Repetition is the key to life, so I'm just going to keep kind of repeating some of this stuff. So transcription, we have the DNA sequence. We open it up. We pair it up with a complementary, so it kind of fits together, RNA sequence that has now the message that gets to be turned into protein. Remember this cheap message that we made? This RNA is able to leave the nucleus. So it will do that. And it will come over to the ribosome, okay? And on the ribosome, it's pairing up with specific molecules called tRNA, okay? And they are dropping off amino acids that match up with the code that they are reading. So we read this mRNA in little 3 base chunks, which we call codons, which makes sense because they are coding, okay? So we pair these guys up kind of like a puzzle piece, and we drop off the correct protein that the DNA was coding for way back in the nucleus, okay? So we have the DNA message. We turn it into a cheap little messenger copy called mRNA. Then the mRNA goes and gets read by these very specific readers that are dropping off the right amino acid to build the correct protein that we coded for. So that protein could be the protein that makes your hair the way that it is, or the protein that makes your eye color the way that it is, or any number of things, okay? But it's getting coded for and turned into that protein based on the original message from the DNA. It just takes a lot of steps to get from point A to point B, okay? So that's just kind of the overarching process here. Any questions about that before we keep cruising? Cool. Usually our sessions won't be this disjointed because we'll just be talking about one little subtopic. But we'll just cover a little bit of everything today, getting pumped for the school year. And if you are a bio nerd like I am, then this is bringing you life right now, which it's bringing me life to talk about all this stuff that I haven't talked about in a little while. Okay, cool. So then the last topic and my last real slide for you is about evolution. And evolution is like the most overarching topic that I can think of for AP biology. It is the most frequent thing tested on the AP exam. So in a week, probably in a week, I'll do like another course intro and I'll break down all of the major topics and what % of the exam is based off of those sections. Evolution tops it off, right? It's 13 to 20% of the exam. So it's really, really, really important. But it's also our second to last unit. And so I'm only going to touch on it briefly today. It's good to know some things to have in the back of your mind because evolution ties into so many wonderful content topics. And it's the best. Like, you can quiz me throughout the year and be like, how does this connect to evolution? And I'm sure I can tell you a way. But you don't need to be super, super highly familiar with all of the specifics coming into it because we have a real long time. We won't talk about evolution until like March. So we have time before we dive into that. And if your teachers are following the same pacing plan as College Board, then you also probably won't hit it until then. So here are the high level things that you should be familiar with coming into the course. So on the left here, this is kind of an example of survival of the fittest, which may be something that you've heard of before. Unfortunately, saying survival of the fittest on the AP exam or in a free response question isn't quite going to cut it. But if you know the rationale behind that terminology, then you'll be in good shape. So all that survival of the fittest means is that in a group of population, there is some way that you look, something that you do. Maybe it's a mating dance. Maybe it's the amount of feathers that you have. Maybe it's that you're a brown mouse instead of a white mouse. There's something that is more favorable than something else for whatever reason. So if we could say in this population, let's say these are mice and these are gray mice. So we have a dark gray, a medium gray, and like a white mouse. Well, perhaps we are living up on a mountaintop, and with global warming, which is very real, and we will talk about it, the snow cap of that mountain is melting. And so the surface of the mountain becomes a deep, dark gray from like wet stone. Yeah, mountains are made of stone. I love coming up with examples on the fly. Okay, so back in the day, the white mice were doing great because they could blend in with the snow. And then the gray mice were doing great because they could find the little parts of rock to hide. But with global warming and all of the ice melting, all of a sudden, those white mice stick out like a sore thumb. And the hawks that like to chill at the top of this mountain are swooping down and eating them. It's an unfortunate reality. Probably could be a true scenario, okay? But what's going to happen is because those white mice are being eaten, our medium and dark gray mice are the ones that are surviving and reproducing. No white mouse chose to become a gray mouse. That's not why we're getting more gray mice. The white mice are just dying and the gray mice are reproducing. And because of that, we see a lot more gray mice on the mountain. Okay, and you can see we're shifting more and more towards this darker gray because now these lighter gray mice or medium gray mice are the ones that are easier to spot on the mountain. So the hawks are eating those, they're dying off, and the darker mice are the ones that have the favorable mutation and they're more likely to survive. Okay, so that's the general process of natural selection in a nutshell with one singular example. Okay, but the important thing to remember is there's no choice in natural selection, right? Students like to sometimes write, like I'll have students write like, the mice chose to become more gray, or the mice started becoming more gray. You can't change your color. I can't decide to become more blonde unless I pay money at the hair salon, I guess, right? But if you can't do that naturally, neither can a silly little mouse, right? So it's the only reason that this is happening is because some mice are dying and some mice are reproducing. And then the other common misconception that sometimes comes from here is that things are just appearing, right? Nothing just comes out of thin air. Like antibiotic resistance doesn't come out of thin air. There's a mutation in a population that then becomes favorable. So that's something that we'll talk about a lot too, but that's like a key concept to understanding natural selection. Okay, so again, just a couple key misconceptions that sometimes students have, okay? And then the only other vocabulary that I'll just kind of throw at you, because it's oftentimes talked about in an intro class, and honestly, like in my AP class, I hardly talk about these things because I just expect students to know them, are homologous and analogous structures, and then divergent and convergent evolution. And these kind of go hand in hand, so I'll talk about them concurrently. So the first thing that we'll talk about is homologous structures. And hopefully you're already recognizing that prefix homo as meaning same, right? From when we talked about homozygous. So homologous, homo, same structure, okay? So homologous structures happen in divergent evolution. So we have some sort of common ancestor, and we have two populations that diverge over millions of years and become two separate things, like elephants and woolly mammoths, which is what you can see in this example. But they stem from a common ancestor. Another example would be humans and monkeys. Humans did not evolve from monkeys because monkeys are still alive and well. We just had a recent common ancestor with monkeys, and then off came humans, off came monkeys, and we're very, very closely related, right? But we didn't come from one another. So that divergent evolution oftentimes leads to homologous structures, where we see structures that are almost identical, but they're doing different things. Okay, so in this example, you can see these purple bones, blue bones, green, red, and yellow are almost identical between a bat and a bird. And that shows some common ancestry, right? Some sort of prior organism that had these bones that then passed them on. And there's also really cool graphics that we'll take a look at that show that that bat wing is also really similar to a human arm, which is also really similar to a whale's fin and a cat's paw. A lot of organisms share very, very similar upper extremity bones, and that's because of a common ancestor. So there was some sort of organism that passed down those structures to now new populations. So that's our example there of the bat and the bird wing. Now on its own end would be convergent evolution. And convergent evolution is when two populations develop a very similar trait, but separately. Okay, they have two different ancestors. They don't share a recent common ancestor. So our example up here is a hummingbird and a moth. Both fly, but they did not come from a recent common ancestor because hummingbirds and moths are way different, right? They both fly because it's beneficial to them. They're off the ground from predators, right? So that's a benefit, but it evolved separately, more probably because of the threat of predators, very much less because of a common ancestor, right? They're not coming from a recent common ancestor. So we call those things like the bird wing and the moth wing, which I have here, insect wing and bird wing. We call those analogous structures. They do the same thing, right? They're like analogies, right? But they're not the same. They do the same thing, but they're not the same. So an insect wing doesn't have bones in it or feathers or blood, maybe a little blood. There's probably some capillaries in there, but it doesn't have all the stuff that a bird wing would have, even though those two do the same function. So homologous structures, same structure, different function. Sometimes, in this case, they're pretty similar, but like a human hand and a bat wing would do different functions. And then analogous structures, different structure, same function. They're doing the same thing, but they don't look the same because they didn't evolve from a common ancestor. So those are some kind of key vocabulary for your evolution unit, which again, may not be for a very long time, in which case you can just keep some of these key points in mind as you're looking at different scenarios throughout the year. Because again, one of AP Bio's favorite things, and AP in general, is to interweave lots of different content. And evolution is a big one that they like to interweave with every other unit. So if you're constantly kind of keeping some of these things in mind, you're setting yourself up for success on the exam. All right, y'all. Well, that, if no one has any questions, that is really the lovely overview of just some things in 43 minutes that you should know going into your AP class. So as I mentioned earlier, we will start like our full educational programming, if you will, on the week of Labor Day, which for a lot of you is probably your first week of school. So September 3rd through 9th, I think it is. That's when we'll start hitting the content heavy, starting with Unit 1. Next week, we may have a session or two on just some other strategies and things to remember coming into AP, so be on the lookout for an email. Otherwise, I'm really excited to continue to work with all of you. And the content will keep coming, and we're going to get really deep diving into AP, and you can ask questions, and it'll be a really great time to learn and grow. So keep coming back and tell your friends, tell your teachers, etc., because we're really excited to work with you this year. Thanks, y'all. Have a great week, and see you next week for some new content.